So next, let us go into membrane potential and diffusion. Till now, we have been talking about how molecules diffuse through the cell membrane depending on concentration difference itself. Now we'll look into how charge of the particular our membrane as well as charge of the particular ions also affect the diffusion rate. So to understand this, first let us first let us look at particular examples of ions. Here we are going to look at two examples of potassium and sodium. So first off, we have a potassium ion, and we have the cell we are going to look at is a nerve cell because that is what we are going to discuss later in later parts of these slides. The assumption that we will take first off is that the membrane is permeable to only potassium ions and no other ions are present anywhere else. Since potassium concentration, as we have already seen, inside the nerve fiber is very high compared to the outside of the membrane, the concentration gradient of the potassium ion tries to push potassium from inside of the cell to the outside, as indicated by the red arrows. So diffusion is occurring along the red arrows from inside of the cell to the outside. Migration of these positive charges, obviously potassium is positive, and movement of positive charge outside creates a charge difference along the cell membrane. Since positive charges are moving out of the cell itself, therefore the outer surface of the cell membrane is now positively charged, whereas the inner surface is now getting slowly negatively charged. Thus, migration of the positive charges generate electropositivity outside and electronegativity inside the membrane. This is very important, as we can see that after some time, this positive charge on the outside of the membrane will try to push potassium ions from the outside to the inside of the membrane again. That is against the diffusion gradient. This is because of the electrostatic force being applied on the potassium ion. As the outside of the cell membrane is more positive, potassium ions are pushed into the cell through the cell membrane into the cell itself. Thus, a diffusion potential is generated. Within a millisecond, this diffusion potential that is generated becomes high enough to prevent further diffusion. Thus, the concentration gradient is now balanced by the electrostatic force that is being applied on the potassium ion. Now, this is a situation of dynamic equilibrium where no more net diffusion of potassium ions occur in either direction. This condition is as I said, a state of dynamic equilibrium, and the diffusion potential is the electrostatic potential that maintains this dynamic equilibrium. In mammalian cell, in mammalian nerve fibers, potassium diffusion potential is minus 94 millivolts. We also have to understand that the diffusion potential sign depends on the charge of the cell membrane on the inside. Because diffusion of potassium was generating a negative charge, that is why the diffusion potential has a negative polarity. And the magnitude is 94 millivolts, which means that if diffusion potential of minus 94 millivolts is applied, then there will be no net diffusion of potassium ions. Next, we move on to another example of sodium ions. This time also we will assume that the membrane is only permeable to sodium ions. But in this particular scenario, the situation is the reverse of the previous one. The amount of sodium or the concentration of sodium ions is way higher in the extracellular environment than inside of the cell. Thus, if the nerve fiber membrane is is permeable to sodium ions, then all the sodium ions will try to get into the cell via the membrane. The diffusion of sodium ions this time generates a diffusion potential again, but its polarity is reverse of that of potassium, which means is that since the sodium ions are diffusing into the cell as indicated by the red arrows, this time around, positive charge is moving from outside of the extracellular space the inside of the cell. Since positive charge is moving along this path, the inside of the cell membrane is now positively charged, whereas outside is negatively charged. Again, we see that an electrostatic diffusion potential is generated, and as before, this finally counteracts the diffusion that was happening due to the concentration difference. Again, thus, the electrostatic potential finally balances the concentration gradient. Thus, within millisecond, diffusion potential again blocks the diffusion of sodium ions. In mammalian nerve fibers, diffusion potential for sodium is plus 61 millivolt. Again, we will see that the charge, the polarity of the diffusion potential depends on the charge that appears on the inside of the cell. So this was the case that happens when we consider potassium and sodium separately, where no other ions are present. Now let us try to quantify the diffusion potential that is present here. As we have seen before in a previous slide, that the diffusion potential is also known as Nernst potential. And at 37 degrees Celsius, which is normal body temperature, the Nernst potential or EMF is given as plus minus 61 into log of concentration inside versus concentration outside of the particular ion. The unit of this EMF is millivolts. This formula is very easy to use, and for specific ions can easily calculate the EMF that is generated, the diffusion potential that is generated due to the concentration gradient of the particular ion. The question arises, what does plus minus mean? The positive or the negative sign depends on the charge inside the membrane when the ions diffuse. That is, this is again, as I was saying, the particular polarity of the EMF will depend on the charge on the inside of the cell membrane after the particular ion has diffused. And that is how the positive or negative is decided. But this simple Nernst potential formula has a very gaping error or a gaping disadvantage that we are not able to cover. That is, it only can calculate the EMF when one ion is present. As we have seen in just before the examples, if only sodium or the potassium ion is present. But this in the real life, the scenario is very different. There are multiple ions whose concentration differ by a lot and whose polarity also differs. But we have to calculate the diffusion potential of a nerve fiber or of any other cell depending on all these concentration polarities, which cannot be given by the simple Nernst potential formula. Thus, if we want to calculate diffusion potential, taking into account several ions, we have to move into a more complicated equation, which is given by the Goldman equation, the Goldman-Hodgkin and Katz's equation. For calculation of diffusion potential for several ions, this depends on polarity of the electric charge of each ion, permeability of the membrane, that is how permeable the particular ion is through the cell membrane. In previous examples, we have assumed that that sodium and potassium are absolutely permeable and there is no hindrance of their movement by the cell membrane. But in this case, we are assuming that there is a particular difference in permeability. And as we will get to know that potassium is 100 times more permeable than sodium. And thus, this particular equation also gives us how this permeability difference plays a role in the overall calculation of the diffusion potential. And finally, the concentrations of the respective ions inside and outside of the membrane are also very important. Thus, the Goldman equation is given right here, which gives EMF again in millivolts as minus 61 into log of concentration of particular ion inside cell membrane into the permeability of the particular ion and summation for all the ions present. Similarly, the denominator of the logarithm is concentration of the particular ion outside of the cell membrane multiplied by the permeability of the particular ion and summation and for all the ions that are present. And thus the equation takes this form, that is for only sodium, potassium and chloride ions, we have concentration of sodium inside the cell into permeability of sodium plus concentration of potassium inside the cell into permeability of potassium plus concentration of chloride outside the cell into permeability of chloride. This we can see there is a difference for chloride itself. This is because the polarity of chloride is the reverse of sodium and potassium, which is positive, and chloride is negative. And thus, we in the equation itself, when we plug in the values, we have to put the concentration of chloride, the concentration of chloride outside of the membrane in the numerator, whereas we'll keep the concentration inside the membrane as the denominator. So this may sound complicated, but it is not very complicated. We will. It is essentially that positive ions have their inside concentration in the numerator, whereas negative ions have the inside concentration in the denominator, and we multiply with them their respective permeabilities. So this was the Boltzmann equation, or the Boltzmann-Hodgkin Cards equation, that is used to calculate the diffusion potential across a particular membrane, or in our particular case, the nerve fiber. Next, we will look into how exactly this membrane potential is calculated in real life. The equation that we have just seen, the Boltzmann equation, gives us a quantitative definition of the EMF. But now we want to actually measure it using a particular equipment. The equipment that is used is known as the microelectrode, which is used to measure the membrane potential of the fibers. The electrode is impelled into the nerve fiber. There is another electrode, which is known as the indifferent or reference electrode, which is placed in the extracellular fluid. This arrangement is connected to a highly sophisticated voltmeter, which is capable of measuring even a few millivolts. The micropipette itself is also special. It has a diameter of less than 1 micrometer and a resistance of greater than 1 million ohms. The reason of this huge resistance is because we are calculating a very small current, a very small. The reason of this huge resistance is that we are going to calculate a very small potential difference across the reference as well as the measuring electrode, and thus this huge resistance helps us to minimize the current that flows according to Ohm's law and easily calculate the value. Here we are, during the whole discussion, you will see that I am going to use different words which are diffusion potential, membrane potential, Nernst potential. All of them are essentially indicate the same quantity, that is the potential that is present on the cell membrane, or essentially the potential difference that is present in the cell membrane across the cell membrane with respect to the outside and the inside. Thus, this is the potential difference that is present across the cell membrane, which is present across the cell membrane. That is what we define as the membrane potential, diffusion potential, and Nernst potential. So this particular device is how we measure the membrane potential practically in real life. Now let us move on to nerves themselves. So nerves have a membrane potential, have different membrane potentials. The first that we are going to discuss is the resting membrane potential. That is the membrane potential that is shown by the nerve when it is not doing anything. No signal is being transducted across it, and it is just resting. The resting membrane potential of large nerve fibers is about minus 90 millivolts. We will look into it how this particular membrane potential is calculated. So there is a huge role of sodium and potassium ions in maintenance of this resting potential. First off, there is active transport via the sodium potassium pump. Remember the sodium potassium pump that we have just discussed a couple of slides back. which actively pumps sodium out of the 3 sodium ions out of the cell in exchange for 2 potassium ions into the cell. The sodium potassium pump continually pumps sodium ions outside cell and potassium ion into the cell. This pump, as we have already discussed before, is electrogenic in nature, as it pumps more positive ion outside the cell and hence generate a negative potential inside the cell membrane, as 3 positive ions in the form of sodium ions are getting out while 2 potassium are getting in, and thus there is a net 1 positive charge that is going outside, and hence the inside of the membrane is gradually becoming the potential of the inside of the membrane gradually becoming more negative. There is also leakage potassium and sodium channels. The protein channels are present through which sodium and potassium ions can freely pass through. There is no gates or any voltage getting on the channels themselves, and sodium and potassium ions can just pass through depending on their concentration gradients. And such leakage channel, such potassium channel, protein channels are known as the potassium sodium leak channels. Potassium leakage to outside the cell is 100 times more, as like a huge amount more, as the membrane itself is 100 times more permeable to potassium than sodium. Thus, given time, 100 times more potassium ions will try to diffuse out of the membrane than sodium ions will try to diffuse into it. So this is again an overview of the whole thing. Here we are seeing the sodium potassium pump with action. Sodium ions are getting out of the cell while potassium are getting into, and the whole process requires ATP, and thus this is an active transport. Whereas leak channels are just open gateways via which sodium and potassium can just pass through depending on the concentration gradient. Sodium tries to move into the cell while potassium tries to move out of the cell, and these are known as the potassium sodium leak channels. These membrane proteins contribute to maintain the following large concentration gradients for sodium and potassium across nerve. Thus, as we have seen, the sodium concentration inside the cell membrane is 14 milliequivalents per liter, whereas outside is 142 milliequivalents per liter. And in case of potassium, the concentration inside is 140 milliequivalents per liter, whereas outside it is 4 milliequivalents per liter. And here, this concentration gradient is actively maintained via the sodium potassium pump, and the leak channels also play a passive role in this maintenance. Thus, the ratios of concentration of these ions from inside to outside are sodium inside divided by sodium outside. The ratio itself is 0.1, and potassium concentration inside divided by potassium concentration outside, the ratio that is maintained is 35.0. Hence, the huge concentration gradient is maintained actively by the cell. Now, we will look into how exactly the normal resting potential originates. So, first, we will look into what is the contribution of the potassium ions. In case of only potassium ions, as we have seen, the ratio of concentration is 35:1. Using the Nernst equation only, the diffusion potential comes to around minus 94 millivolts. Thus, if only potassium ions were present, the resting membrane potential would have been minus 94 millivolts. When we next look into the contribution of sodium ions, the ratio of the sodium ion concentration is 0.1. Plugging which into the Nernst equation formula gives us the diffusion potential to be around positive 61 millivolts. But when potassium ions also come into play, when potassium ions but the situation changes when we consider both the effect of sodium and potassium together. In combination with potassium ions, the resting potential is calculated using the Goldman equation. As the permeability of potassium ions is 100 times more than that of sodium ions, the final diffusion potential or the membrane potential lies hugely in favor of potassium, and which after putting into in the values comes out to be around minus 86 millivolts. The as we have all, as I already said, this huge negative membrane potential is because the permeability of potassium ions being 100 times larger than that of sodium ions, and thus the membrane potential is way more towards the potassium ion side than that of the sodium ion side. Since we have seen in the first concentration diffusion table, the concentration gradient table, the major players in determining the membrane potential is are the potassium and the sodium ions. This is because of the huge concentration of the potassium and the sodium ions present in the intracellular environment and the extracellular environment. As we have seen, if you just refer back to it, they are the only ions that are present in such huge quantities, whereas other ions such as calcium, magnesium, phosphate are present in way lower quantities. And thus, these two ions are the major players which determine the membrane potential. Next, we have seen the sodium potassium pump come up again and again. It also makes an appearance here. The sodium potassium pump further contributes to the resting membrane potential. As we have seen, the pump is electrogenic in nature. That is, it actively contributes to changing the charge across the membrane. An additional loss of positive ions is generated, created by the electrogenic pump, and which causes an additional negativity inside the membrane. This negativity accounts for around minus 4 millivolt, accounts for around minus 4 millivolt difference. Thus, combining the minus 86 millivolt due to sodium and potassium, as well as the minus 4 due to the sodium potassium pump, we have the final resting potential as minus 19 millivolt. Next, let us look into nerve action potential. We are done with the resting potential of nerves, and we are going to talk, change gears and talk about how exactly nerve conduction takes place. Nerve conduction takes place via a phenomenon known as action potential, which is a rapid increase in the membrane potential of the nerve. This increase in membrane potential is how nerves conduct signals from one part of the nervous system to another. Rapid changes in membrane potential along the nerve fiber is known as action potential, and this is responsible for transfer of nerve signals. Action potential has three separate stages, as we can see from the diagram here. The first stage is the resting stage, where the membrane potential is the normal minus 90 millivolts that we have just calculated. In this stage, the nerve is said to be polarized. Then the next step is the depolarization state. The membrane becomes suddenly permeable to sodium. This permeability increases way higher than the base permeability of sodium. And thus, sodium ions diffuse in large quantities into the cell, which neutralize the polarized minus 90 millivolt of nerve fibers. As we have seen that the membrane potential is minus 90 millivolts because the inside of the membrane is at a negative potential. But the huge influx of positive ions in the form of sodium ions neutralizes this negative potential, and thus the overall membrane potential rises, and this is known as depolarization. And thus this is the depolarization part of the whole action potential plot. Finally, we have the repolarization stage. At a particular point, the sodium influx increases the membrane potential to around plus 35 millivolt, which is given as the overshoot. Once at this point, within 10,000 of a second, the sodium channels start closing. So sodium channels start closing, which means the influx of sodium ions has stopped. Simultaneously, there is an opening of potassium channels. As we have seen, the potassium gradient is outside, that is opposite of the sodium. And the opening of the potassium ions causes huge amounts of potassium ions to exit the cell. This exiting of the cell essentially is the movement of positive charge outside of the cell, which again polarizes the membrane by generating a negative charge on the inside of the membrane. Thus, the movement of potassium ions and simultaneous closing of the sodium channels allows the nerve fiber to be repolarized, and thus this stage is known as the repolarization stage. Rapid diffusion of potassium ions to the exterior brings the membrane potential back to the normal resting potential, and this is called repolarization. And after depolarization, there is a slight overshoot below the initial resting condition, and finally it returns to the initial resting membrane potential. And this whole phenomenon of resting of action potential is thus divided into the resting stage, depolarization stage, and repolarization stage. And as we can see, that the whole phenomenon occurs in a very small time scale as well. Moving on, let us now look at exactly how this action potential is generated. There are two main players in the generation of action potential, which are the voltage gated sodium channels and the voltage gated potassium channels. As we have discussed just some slides before, voltage gated protein channels are protein channels whose opening and closing depend on the overall membrane potential. So if the membrane potential changes, the opening or the closing of the voltage gated channel is affected. The voltage gated sodium channel the opening and the closing of the voltage gated sodium channel depends on the membrane potential. It has two gates: the activation gate near the exterior and the inactivation gate near the interior, as you can see in this diagram. This is the activation gate and this is the inactivation. When first off, let us discuss the activation of the sodium channel. As membrane potential rises from -90 to 0 millivolts and reaches first around -7 to -50. So there's a gradual rise from 0 to -90 as -90 to 0 as we just see. And when the membrane potential reaches around -70 to -50, a sudden conformational change opens the activation. So now the activation gate is open, and initially the inactivation gate was also open. Thus, there is now an open path for the sodium ions to come into the cell, and hence sodium ions permeability shoot up to 500 to 5,000 fold the higher than the normal baseline. So huge influx of sodium ions is seen, which is this activation state. Next, there is an inactivation state of the sodium channel. The initial increase of the potential that opens the activation gate also closes the inactivation. But this opening of the activation gate and the closing of the inactivation gate has a time lag of a few 10,000ths of second, which means that a huge influx of sodium ions are allowed, but the initial stimulus also then blocks the excess entry of sodium ions into the cell. Thus, the initial increase in potential first activates the channel, allows influx of sodium, huge amount of sodium ions, and then inactivates the channel by closing the inactivation, which stops the sodium ion flow into the cell and begins the repolarization process. This is the inactivation state that is shown right here. Next, let us look into the action of potassium, voltage-gated potassium. Again, voltage-gated potassium channels are channels whose opening and closing depend on the membrane potential. The potassium channels exist in two stages: the resting stage and the activated stage. During the resting stage, potassium ions cannot pass through the channel, whereas in the active state, the potassium ions can pass through the channel. As the membrane potential gradually rises from minus 90 millivolts to zero, the potassium channel is also activated. But the potassium channel has a delay in activation or activates very slowly. Once activated, the potassium ions allow a huge diffusion of potassium ions from inside of the cell to the outside. That is the opposite of the sodium gradient. But because of the delay, the potassium channels only open when the sodium channels are again closing back due to inactivation. Thus, there is a simultaneous influx of simultaneous. This would be exit of sodium potassium ions out of the cell along with decrease of sodium entry into the cell, which combine to repolarize the nerve fiber. So there is the inactivation of sodium gate, which doesn't which prevents further entry of sodium ions, and secondly there is opening of the delayed opening of the potassium channels, with which allow the flow of potassium ions out of the cell, which both of them together allow the repolarization process to start. So this is a diagram representing the conductances as they occur during the action potential. So first off, this is looks a bit complicated, but as I will try my best to explain everything that is going on. So first we will look at the diagram that is up above. So the first this diagram. So this shows the action potential that is up along with the membrane potential as a function of the membrane potential. So as we have seen, this is the resting stage followed by the depolarization, overshoot, and the repolarization. So this is it. Next, we'll look at the ratio of conductances of sodium and potassium. So initially, we have a huge the conductance of potassium ions is way larger than sodium by because the permeability of potassium ions is 100 times more than that of sodium. And this in the resting phase, resting stage, the conductance is actually contributed to by the leaky channels that we have seen. So the whole, if we see the ratio is very much in the decimal side, which means that the denominator is way larger than the numerator, which is obviously the case as the potassium conductance is way higher than the sodium conductance. But as the action potential starts, we see a gradual increase in this ratio, which means the numerator is increasing gradually till it reaches a maximum, which is way higher than the one unity, which means that the sodium conductance has gone way up than the potassium conductance. But then we again see during the repolarization phase, the denominator increasing while the numerator is also decreasing, as inactivation of the sodium channels prevents further conductance of sodium, and opening of the potassium channels allow further conduction, allow the further conductance of potassium, and thus the denominator is increasing while the numerator is decreasing. And we see that the whole conductance ratio plummets down to a very low value, which is below even the resting potential value. And after which it then stabilizes back to the initial resting membrane conductance ratio. This was the above graph. Next, let us look at the conductance of sodium and potassium individually. The conductance of sodium, let us look at first, is way lower than that of potassium in the resting phase, increases by a huge lot in the depolarization stage, and then again plummets down during the repolarization stage back to the resting membrane potential levels. Conductance of potassium is already, as we can see, way higher than that of sodium, and thus the graph starts hugely up in the y-axis. As we can see that potassium conductance does not increase till around the point that the potassium sodium conductances have become either starting to dwindle or have reached their maximum. That is the point at which the potassium conductance actually starts to increase greater than its resting value. And as the potassium conductance increases, the sodium simultaneously decreases. Potassium gains a peak. Potassium conductance gains a peak, after which potassium channels also start to close, which also drop the conductance back to gradually back to the initial resting levels. This was the conductances of sodium and potassium during the action potential. So now, after we have looked into what exactly action potential is, we will look at various characteristics of the action potential. First we will look at the initiation of the action potential. A positive feedback cycle is responsible for the opening of the sodium channels and initiation of the action potential. As nerve fiber has as the membrane potential of the nerve fiber rises just by a little bit, many sodium channels open, which causes further depolarization, which opens more sodium channels. This cycle continues till all voltage gated sodium channels are activated. What does this mean? This means is that, as we have seen, the sodium channels, the voltage gated sodium channels open once the membrane potential reaches around minus 70 millivolts. So thus, once there is a small rise in potential, many sodium channels open due to the voltage gated channels opening, but this further causes a depolarization. There is further increase in membrane potential, and this triggers further opening of voltage channels along the nerve fiber membrane, and hence the one by one all the sodium channels that are present across the membrane are open. Action potential will be initiated with the initial rise in voltage. The initial rise is essential. This initial rise in membrane potential needs to be greater than a threshold in order to initiate an action potential, which means that since we have seen that the first voltage gated sodium channel starts to open around minus 70 millivolts. So if our nerve fiber has an increase in membrane potential, which is very low and not enough to cross that minus 70 millivolt threshold, then the action potential will not be generated. An initial membrane potential rise to around minus 65 millivolt is required to open enough sodium channels to begin the vicious cycle of the positive feedback loop. The direction, next let us look at the direction of propagation. Action potential travels in all directions along a uniform nerve fiber until the entire membrane is depolarized. So if the nerve fiber is uniform, the action potential will travel in all directions uniformly. Finally, there is the all or nothing principle. Once the action potential is initiated, either the entire membrane will be depolarized if the conditions are right, otherwise the depolarization wave does not travel at all. As I said, if the threshold is crossed and the conditions are optimal for the action potential to occur, then the entire nerve fiber will be depolarized. But if the threshold is not crossed and the conditions are not optimal for action potential generation, then there will be no depolarization of any part of the nerve fiber, and thus there will be no action potential generated. Thus the final verdict is, if action potential occurs, it will occur for the entire nerve fiber and depolarize every part of the nerve fiber, and there will not be any localized action potential occurring on the nerve fibers. Next, this particular diagram shows the direction of propagation. Even if the action potential is generated some way off toward the right of the nerve fiber, the action potential travels in both directions uniformly, as we can see, and it travels along the nerve fiber till the entire nerve fiber is completely polarized, completely depolarized. Next, let us look into how about specifics of the nerve signal transmission. To understand nerve signal transmission, First, we have to understand the characteristics of the nerve fiber itself. Nerve fibers are large nerve fibers have a layer of myelin sheath covering. The myelin sheaths are insulating material that cover a large nerve fibers, as we can see here and as well as here. Whereas smaller nerve fibers are unmyelinated and are twice as numerous as myelinated ones. The central core of the nerve axon is obviously the larger extension of the nerve fiber body, neuron body that helps in transmission or the sending of the signal from the neural body to the next neuron. And the other part of the other two parts of the neuron include the dendrites, which receive this signal, as well as the cell body, which contains the nucleus of the nerve cell. The central core of the nerve axon is surrounded by this sheath, which is several times thicker than the axon itself. As you can see here, the axon is small as compared to the huge myelin sheath that is covering. The myelin sheath acts as insulation and prevents flow of ions. So wherever the myelin sheath is covering the nerve fiber itself, there won't be any flow of ions through it, and thus there won't be any depolarization at that part. And the myelin sheaths are produced by specialized cells known as Schwann cells. Schwann cells are shown right here, and this is how they are present in Schwann cells are shown right here, and this is how they are present in the actual nerve fiber. And the neurons are present like this along the membrane of the Schwann cells. About every 1 to 3 millimeters along the neuron, the fiber is devoid of any myelin sheath. That is, there is a gap in the myelin sheath, and this is known as the node of Ranvier. Node of Ranvier plays a very important role in the nerve signal conduction. We will see in the next slide. So, as we have already discussed, no ions can flow through the myelin sheath. Thus, the action potential occurs only at the nodes of Ranvier. Thus, the current which flows into the nerve fiber and out of it from the surrounding extracellular fluid to the axoplasm of the cytoplasm of the axon, it excites successive nodes one after another, meaning that one node of Ranvier will be excited, and only the other node of Ranvier after it can be excited, as the middle portion has myelin sheath all over it. So one node will be excited, after which another node will be excited, after which another node will be excited. This is known as the jumping conduction or saltatory conduction in myelinated fibers. The depolarization jumps along the nerve fiber, increasing the velocity of transmission by 5 to 50 fold. So the nodes of the depolarization occurs only at the nodes, and it looks as if the depolarization is jumping along nerve fiber only at this point, then it jumps to the next node, then it jumps to the next node. This increases the velocity of transmission by 5 to 50 fold, because the whole nerve fiber in middle does not need to be depolarized, only the nodes need to be depolarized. Further, much less energy is required to repolarize the membrane, as we have seen that after depolarization, repolarization also occurs, and repolarization will occur because of the movement of ions across the membrane. And this movement of ions, obviously, to maintain the concentration gradients across the membrane, we have to the major part is played by the sodium potassium pump, which starts pumping the excess sodium ions that have come into the cell and the excess potassium that has gone out of the cell in the reverse direction, so that the concentration gradient can be re-maintained. And this, obviously, as we have seen, requires ATP, and thus much less energy is required to repolarize the membrane in case of myelinated nerve fibers, as ions are lost only via nodes. Only this portion itself loses the ions, not the whole nerve fiber membrane. And thus 100 times less ions need to be trafficked across the cell membrane to re-polarize or re-maintain the concentration gradients across it. Then otherwise would be necessary if the whole membrane is depolarized. So that was all about signal transaction. We'll look into the refractory period about action potential. We come back to the scale of a single unit. New action potential cannot be initiated till the membrane is repolarized. This is obvious, as we have seen the once the depolarization wave takes the membrane potential up to about plus 35 millivolt, we have to reach for back to the minus 90 millivolt resting membrane potential before another action potential can be generated. And thus, till the membrane is repolarized, no new action potential cannot be generated. Further, sodium channels, after being inactivated, will not be activated however much the excitatory signal is applied. So there is a second point that we have to encounter here, which is that the sodium channels that we have seen have two gates. One is the activation gate and there is the inactivation. Say we are trying to forcibly open the sodium channels and try to make it the nerves, the neurons membrane potential rise. So as we have seen before, around minus 70 millivolts, the activation gate should open. Even if the activation gate opens, because the inactivation gate is still closed, thus the sodium ions cannot get into the cell. And as sodium ions cannot get into the cell, next action potential will not be generated. So what does the whole gist of the thing means? That once inactivated, the inactivation gate is not going to open. If the inactivation gate does not open, sodium channel ions cannot pass through and cannot initiate a second action potentials just after the ending of the first. Only after a fraction of a second, the membrane repolarizes. The repolarization process is very fast, and thus the membrane reaches back to the resting membrane potential within a fraction of a second. And once the resting membrane potential is reached, the sodium channels can be reactivated as the inactivation gate again opens up once the resting membrane potential of minus 90 has been reached, and then a new action potential can be generated. Thus, we have only have to wait for a fraction of a time, which is known as the refractory period, before the another action potential can be generated. The period within which no action potential can be generated is known as the absolute refractory period, and this is around 1.2500 of a second in large myelinated sheets, large myelinated fibers. Finally, I would like, this was all we had to talk about membrane physiology of the nerves themselves. About nerve conduction, we will look into at separate lecture down the line. The next lecture will be on muscles and the membrane physiology of the membrane muscles themselves. For the references of these lectures, I will try to guide you towards the chapter 4 and 5 of textbook of medical physiology by Arthur C. Guyton, the 11th edition. Finally, I would like to thank you for listening to this video.